Check one, check one. Okay. Right, people. Sorry about that. Yeah. So, BioEve paper 29. Paper 29. We're going to discuss that. I hope you have answered the paper. Lamai, did you answer the paper? Answer karada. Nadda. Answer karane. Very good. Right. So let's see. Yeah, answer me. Did you answer the paper? Yes or no? This one. 29th one. Yes, sir. Yes, sir. 3 bags full, eh? Yes, sir. Yes, sir. Answered the paper. 29. Right. So I have given you 2 weeks to answer. Right. So you have to find some method to work regularly. We'll try to regularize all these work. And then balancing is more important. Eh? Your duties you have to achieve. Excuses are not important. Eh? So it's raining. I lost my tooth. I couldn't come for the class. I didn't know. Right. I thought that's a new tooth. So many things you can say. Ne? Right. Only very few people can say that I did it. I had all these troubles, but I did the work. Very few people can say that. Those people, they are great. Right. Moving to the first question. Moving to the first question, the correct statements are the few non-living entities show one or more living characteristics. They are asking correct one. Ne? A few non-living entities show. A few non-living entities show one or more living characteristics. So what do you say about that? Right. Few non-living entities. What they say, few? They're asking correct. I think the resource book has given many. Resource book has given many. So few and many is giving a different meaning, isn't it? So statement A wrong. From molecular level to the biosphere, there is an order and organization in organism. Second is order. Okay. All organisms begin their life as a single cell, correct? Growth and development are two consequent actions that happen in the lifespan of organism. Growth and development are two consecutive actions. That's correct, Nathan. Action and processes are same. Action and processes. Do you see any difference? I don't feel any difference. Growth and development are two consecutive actions. Processes. Actions and processes. Is there a difference? Action? Processes? Slight difference is there. Eh? Action process is something which happen continuously. Action. I can't take anyway the statement D as wrong. I can't take that. Right? So happen during the lifespan of an organism. Right? So I'm taking that as a correct statement. What do you think about it? Right? And growth and development are two consecutive action that happens. So how about taking A, B, D as answers? A, B, D. I'll take A, B, D. A, B, D. A, B, D. Fifth one is the answer. A, B, D. Fifth one. Question number 2. Consider following statements about main organic compounds of organisms. Main organic compounds of organisms. Oops. Main organic compounds. Right. Consider following statement about main organic compounds of organisms. Right. Main organic compounds. They're asking people Consider the following statements about main organic compounds. The incorrect statements. Incorrect one they expect me. Amylopectin is a branch form. Amylopectin is a branch form of polysaccharide. Amylose and amylopectin. Amylopectin branch is correct. And then inulin stores sucrose as energy source in dahlia. Inulin stores sucrose wrong. Neither. No, we can't agree with that. And then keratin is present in component in the cell wall of fungi. Wrong. Neither lamai. Exoskeleton of arthropod keratin, not keratin, chitin. Consumption of excess saturated fat and trans unsaturated fat contribute to atherosclerosis. Excess saturated fat and trans unsaturated, correct. So I'm settling people here with answers. What they ask, following main organic compound. Incorrect statements are incorrect ones will then B and D are incorrect. B and D. Where is that? B and where is that? B and C, ne? B and C. B and C. Second one. B and C wrong. Second one is the answer. Right? Second is the answer for that. Right. Moving to third question. Moving to third question. Below are some steps of experiments conducted to identify non-reducing sugars. They are asking correct order of the above steps. Correct order of the above steps. So how do you take that? Neutralizing the medium with sodium bicarbonate, add few amount of Benedict's solution and boil. So what are the order, people? Add dilute HCl and then boil, add few Benedict solution. Right. So what is the order here? Right. CAB. Either CAB, CAB cab. Fourth one there. Dilute HCl and then boil. So it hydrolyzes. Ne? Then neutralizing should be done next. After that, you can add the bending. So CAB correct. So I'm taking fourth one as the answer. Fourth one as the answer. Next one, people, question number 4. Question number 4. The correct statement about subcellulars. The correct statement about subcellular component is. Correct statement regarding subcellular component is lysosomes, enzymes in lysosomes, enzyme in it catalyzes breakdown of hydrogen peroxide. Wrong, neither. Lysosomes. So this is actually done by peroxisomes. No. Chloroplast, circular DNA, 70S ribosomes and many enzymes. Phosphate granules. Wrong. Phosphate granules are present in people. Where do you find phosphate granules? So in the test for non-reducing sugars, we first do Benedict test and see there is any reducing sugars. And then do it the normal test to confirm the presence of non-reducing sugar. Yeah, that's correct. That is correct. But how it is affecting this one? Answer. Does it affect the answer here? Below are some steps of the experiment conducted to identify non-reducing sugars. Sairi, what you say is correct, but I don't think that is affecting for the order here. Sorry. I don't think it's affecting the order. What do you say then? Add few amount of Benedict and then solution and then boil B and then add dilute HCl and then neutralize. I don't think it's set, right? Because it says already below are some steps of experiments conducted to identify the non-reducing sugar. Non-reducing sugar. The correct order of the above steps. Right. Since you're asking that question, it doesn't affect. But anyway, shall we check the practical syllabus to see whether something is there? Test for reducing sugars. Test for non-reducing sugars. Add 2 ml sucrose solution to Now look at this summary. It says at 2 cubic centimeter here. Practical syllabus part of practical syllabus content of the practical book. It says add 2 cubic centimeters of sucrose solution into 1 cubic centimeter of dilute HCl, boil, neutralize with bicarbonate, and then carry out Benedict test. That is how it goes. So I think still what we have taken is valid because they have already mentioned it's a reducing sugar. So it's not like that we try to find out. It's already mentioned that there is a reducing sugar. Now, if they had not mentioned it, suppose it is water and sucrose, you want to distinguish, ne? Then something like that. You don't know what it is. The unknown solutions. You do test it directly, no answer. Then you add the HCl, hydrolyze it, and then try. Then you get the answer. If you don't get answer for both, then you know people reducing sugar as well as non-reducing sugar, both absent, right? Sir, can you speak bit louder? What's wrong? You can't hear? Ukshika, I think it's your one. You have to increase your volume. Can hear other people can hear me very well. No, sir, we can't hear. Eh? Khansa, Hakim. Really, you can't hear? I don't think so. This is okay. Yeah, sir, can't hear. It is so low. Okay, I don't know what. Wait. Let me see. I don't know why. How about it now? Is it okay now? Is it okay now? Is it okay now? Yes or no? Okay now or not? No. Hello. I don't know why it happened. Right, moving people to next question. I don't think it's the issue. Right. The correct statement about so what's the question we did last? We did the fourth one. We were discussing fourth one. The lysosome enzymes. Right. Check the fourth one, people. Lysosome enzymes first is wrong. Chloroplast, circular DNA, 70 ribosome, phosphate wrong. Then glycosome convert amino acid into sugar. Wrong, neither. That's wrong too. And then rough endoplasmic reticulum produce transport vesicles. That is transport vesicles. Correct, neither. Secreting vesicles for Golgi. Golgi apparatus produce peroxisomes. Wrong. So we have the answer. We can go with the fourth one as the answer. Right. Question number 5. Question number 5. Given below are some steps of meiosis. The correct order of the above steps are some correct steps of meiosis. How to take the correct order? Crossing over take place, formation of chromosomes with two sister chromatids. Generally, genetically non-identical two haploid daughter cells are formed. And then sister chromatids move towards opposite poles. Homologous chromosomes arranged randomly at the metaphase. How do you take the right order? Formation of chromosome with two sister chromatids. Genetically non-identical haploid. Right, crossing over. Homologous chromosomes arranged randomly at the metaphase plate. So you can't take that. Crossing over take place. This is first. Then come here. Homologous chromosomes arranged randomly. Then sister chromatids. So this is number 2. Sister chromatids move towards opposite poles. Genetically identical two haploid two daughter nuclei are formed. Formation of chromosomes with two sister chromatids. Wrong, neither. Try that again. So what is your answer? Right, we'll take first one, people. Crossing over take place. Then homologous chromosomes are arranged randomly at the metaphase plate. Homologous chromosomes. So that's 2. Then genetically identical two daughter homologous chromosomes arranged randomly at the metaphase plate. Formation of chromosome with two sister chromatids. Chromosomes. Chromosomes with two sister chromatids. Genetically identical. What is your answer, people? What is the answer you have received? Formation of chromosomes with two sister chromatids. Chromosomes with two sister chromatids. We know, people, this says meiosis. So chromosome with two sister chromatids. This is happening in prophase. Prophase, you can see that the chromosomes with two sister chromatids are going to form. Genetically identical, non-identical haploid two daughter nuclei are formed. Chromosomes with two sister chromatids. Suppose we take this one as first event. B. Crossing over. A. Homologous chromosomes are arranged randomly. Then E. And then homologous chromosomes are arranged randomly. Genetically non-identical haploid two daughter cells. C. And then D. So is it B, A, E, B, A, E, C, D? B, A, E, C, D. 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 B, A, BAE, BA, BAECD. Do you have that? BAE, DCCD, BA, BA, BAE. So B is propase 2, C occur. BAE, DC. So BA. Yeah, we'll check that name. Right, check those statements again. Right, we'll see the phase neither. Yeah. Crossing over take place. What is that, people? Propase. Propase 1. Okay, propase 1 crossing over. Formation of chromosome with two sister chromatids. Formation of chromosomes with two sister chromatids. Can't it be G2 phase? Formation of chromosomes with two sister chromatids. And some people say propase 2. Yeah, propase 2, propase 2. Formation of chromosomes with two sister chromatids. So it is in the propase 2. Okay, you have to then check the book. Um, some people are highlighting and then showing it to me. Okay. Genetically non-identical, the rest of the part is okay then. B position is a issue, ne? Then, then shall we try to, because we don't have this order. So, how it goes then? Crossing over, homologous chromosomes are arranged. Genetically non-identical haploid two-dot nuclei are formed within one cell. Then B, ne? And then D. So then A, E, A, E, C, B, D. Do you have that? A, E, C, B, D. Oh, there, ne? Third one. And then we'll take it as third. Huh? Then I'll take the answer as third. Question number 6. The correct statement. So both propase 1 and both propase 2 chromatin condensation to form chromosomes which are connected by two sister chromatin. No, that is, shall we check that? I didn't check the resource book for that. Let's check it since you have a small issue. Propase 1. It says cell enters the propase from the interphase, chromosomes begin to condense. Next, zipper-like structures in endodermal complex during the synapsis. Non-sister chromatids paired homologous break this process crossing over. It's not mentioned here. Now it happens. Now you know people in the propase, the sister chromatids chromosomes condense, right? No, sorry, there is a small issue, right? Propase sister chromatids are become visible people after crossover, after crossing over only, right? Now it's not given, but we know people there is a difference compared to the mitosis. What is that? The sister chromatids are not becoming visible at the initial stage of the propase 1 that we discuss, I think, right? They become visible after the crossing over, after crossing over. But then there is chiasma formation, right? So I think then formation of chromosome with two sister chromatids, we have to then take for the propase 2. Then, yeah, it says propase 2, nuclear break during this produce chromosome with two sister chromatids, right? So they have mentioned it there. So if you try to right, go to the resource book, then the answer is going to be third one, right? Why can't be propase 1 event? Propase 1 event, sister chromatids become visible after the crossover. Next thing is it become after the crossing over, right? So I think this answer is good, right? Moving people to next question, question number 6. Question number 6. Question number 6. Question number 6. The correct statement regarding tumor, cancer, and gall is: cancer cells are always do not respond to the body control mechanism. Cancer cells always, it says that cells always do not respond. Wrong, neither. Cancer cells do not need growth factors at all. Wrong statement. Ne? They're asking correct one because they need growth factors, but they don't. They can produce their own growth factors. And then the plant cell differentiate is controlled by hormones such as auxin and cytokine. Plant cell differentiation is controlled by auxin. I think this is C is okay. Auxin and cytokinins. Plant cell differentiation is controlled by hormones such as auxins and cytokinins. Differentiation is controlled by auxins and cytokinins. Differentiation is controlled by. When forming galls, plants produce undifferentiated. Well, what is given the plant cell differentiation? Differentiation controlled by hormones such as auxin and cytokinin. Auxins and cytokinin. What is the word given if it is not differentiation? Plant cell differentiation. Development. Is it given development or differentiation? Plant cell division, not differentiation. No. Division. Okay. Plant cell division is controlled by. So then it's wrong. Eh? When forming galls, plant cells produce undifferentiated mass of cells. Correct. Most of the malignant tumors can be completely removed by surgery. Most of the malignant tumors can be completely removed by surgery. What they say, people? Most of the malignant, what they say? Benign tumors. Ne? Wrong. So therefore, the answer is fourth one. Answer is fourth. Answer is fourth. Right. Next question. Question number 7. The question number 7. The correct one from the below statement. Correct one from the below statement is. Right. Check them, people. RNA molecules which act as early enzyme did not have the ability to replicate. They are asking correct one. Neither. RNA molecules which act as the early enzymes did not have the ability to replicate. Wrong, neither. RNA had the ability of replication. So first statement is wrong. Lipid contributed to the growth of the protocells. Lipid contributed to the growth of the protocells. Lipid bounded vesicles. Crustaceans were the first group of animals to colonize the land. Crustaceans. And then people first Earth atmosphere was oxidizing one. That's wrong, neither. We said that the early atmosphere was a reducing one. Wrong. And then at present, the diversity of prokaryotes is higher than that of the eukaryotes. Diversity of prokaryotes are higher than that of the eukaryotes. So if you take here the statement, this is wrong. Eh? This is also wrong. Crustaceans, insects, and spiders. This is correct. Wrong. So we'll take people second one as the answer. Question number 8. The correct statement regarding artificial classification is A. Correct statement regarding artificial classification is A. Evolution relationships are considered. Evolution relationships are considered. Artificial classification. Evolution relationships are not considered. Ne? First statement is wrong. Then based on many characteristics. No, few characteristics. Wrong. The next one, people, easy to use. Easy to use, that is correct. Easy to use, easy to classify. Yeah, we can agree with that. Right? Easy to use, yes. And then we will develop after study of evolution. No. No. So what are the answer, people? What they ask? Correct one. Then C only. Correct one, C only. Other statements, they are wrong. So we have the answer, third one. Question number 9. Question number 9. Question number 9, the incorrect statement about binomial nomenclature is incorrect statement regarding binomial nomenclature. System accepted worldwide. Binomial nomenclature, system accepted worldwide. Correct. Made up of Romanized words. The Latinized. No. Combination of generic name and specific epithet. Yes. Only the first letter of generic name capitalized. Yes. Third word can be used to represent the subspecies or a variety. Correct. So we have the answer. Second one is the answer. Question number 10. The incorrect one. The incorrect one from the below combination is Ernest Haeckel introduced the kingdom Protista. Correct. Linnaeus classification of flowering plants. Linnaeus. Correct. Theophrastus classification of plants according to the habitat. Theophrastus Harini. Aristotle introduced plants and animal kingdom. No, kingdom, he didn't use that word kingdom. Kingdom came from Linnaeus. Right? Kingdom came from Linnaeus, so we can't take that. Then Carl was introduced the three-domain classification system. Yes, so we have the answer, fourth one for question number 10. And then people, we are moving to question number 11. Question number 11. The correct order, one of the organism with the contractile vacuole, foot vacuole and eye spot respectively. What is your answer, people? What is your answer? Fifth one. Aisha says fifth one is the answer. Sayuri also fifth one. I think fifth one. All of you say fifth one. That is the answer, people. I'm agreeing with your answer. Fifth one. Amoeba, Paramecium, Euglena. Right? Contractile vacuole, amoeba, and then foot vacuole, Paramecium, and eye spots are present in Euglena. Question number 12. Question number 12. The correct statement regarding plant cell. Plant cell is question number 12. Meristematic cells have thin cytoplasm. No, we said not thin, the dense cytoplasm. So wrong statement. Dense. Guard cells do not have chloroplast. No, they have chloroplast. So do not have is wrong. And then the parenchyma cells are found just below the epidermis of the young stems. Parenchyma present just below the epidermis, yes. And then sclerites become living at the functional maturity. Sclerites become living, no. They're dead cells. Tracheids are present only in vascular seed plants. Wrong statement. So we have the answer, people. What is the answer? Third one. 12th question, third is the answer. Question number 13, the incorrect statement regarding water potential is. Incorrect statement regarding water potential is. Incorrect about water potential. What is your answer for question number 13? Any system that contains water has water potential. Correct. Right. And then if the cell is placed in pure water, there won't be water movement after considerable period of time is passed. If the cell is placed in pure water, there won't be a water movement, wrong. There won't be a net water movement. So there won't be a water movement, wrong. It has to be net. No, wrong. Water moves, but no net water movement. They're asking incorrect. Turgor pressure contribute directly or indirectly for the support of woody as well as non-woody plants. Not for woody neither. Turgor pressure contribute directly or indirectly for the support of woody as well as non-woody plants. What do you say about that? Right? Wrong statement. What is given in the resource book? Can you check it there? Right? Turgor pressure. I think this woody is wrong. Woody is wrong, I think. So, no. There can be both positive and negative pressure potential in some plants. negative and positive pressure some plants, tissues, which are mainly contained it's they can be negative pressure, yeah, root pressure. You can know people in the xylem, positive pressure and negative pressure both can exist, right? Root pressure under that, right? Positive, otherwise negative, ne? Correct. Then water potential is related to potential energy of water molecules. Yes. So we have the answer. I'm taking people the answer as two answers, second and third. Second and third, right? Because second also has some issue, ne? So it has to be net water movement. Question number 14. Question number 14. The incorrect statement is the incorrect statement. The energy is not needed for the upward movement of water in the xylem. Correct. Weak bonds formed between the water molecules and xylem vessel walls help to maintain water column within the xylem. Weak bonds formed between water molecules and xylem vessel wall help to maintain water column within the xylem. Weak bonds between water molecules and xylem vessel walls help to maintain. Correct me? What are those people? Weak hydrogen bonds and then addition, cohesion. And then guttation is, this is addition. Guttation is observed when root pressure is higher than the transpiration pull. Guttation is observed when the root pressure is higher than the transpiration pull. Correct. And then sugar source is sugar producing organ. Sugar source is a sugar producing organ. Can be, ne? During phloem unloading, sugar is moved towards the sink as well as water moved towards the xylem from the phloem occur through osmosis. Sugar movement wrong, ne? The osmosis fifth statement, right? So I think we can end up here the fifth one as the answer. End up with the fifth one as the answer. Question number 15. Question number 15. Right? What is your answer for question number 15, students? Right? The incorrect statement is xylem vessels are absent in softwood. Xylem vessels are absent in soft. What is softwood? What is softwood? Can somebody tell me what is softwood? Softwood is the wood present in the gymnosperm, right? Softwood. What is hardwood? HARD. Hardwood is the angiosperm wood, right? Softwood and the hardwood. And then people, softwood and hardwood. So 14th question, the resource book says a continuous water column is formed due to unusually hydrogen bonds. Yeah, okay. So hydrogen bonds, right? Well, cohesion, addition, cohesion, both you can take as hydrogen bonds. They are hydrogen bonds, right? Any issue, Sairi? You can take it as hydrogen bonds, ne? I didn't want to read that part anyway. Weak bonds formed between the water molecules and the xylem vessels walls help to maintain that. Statement wise, correct? Okay, no? Right? Correct, ne? So hydrogen bonds are stronger bonds. Ne? Wrong. Why do you say hydrogen bonds are stronger bonds? Now they are stronger people when they come to the solid stage, right? Otherwise, in the liquid stage, people hydrogen bonds are weak bonds. You want me to check that? You want me to check it? Right? We will be very careful in answering you know, words word struggle. What is this? I'm going to read people ascent pressure, radial transport, upward movement. These xylems have cohesion of water molecules transmit. Cohesion tension hypothesis. Negative pressure potential helps under tension according to cohesion. Addition and cohesion facilitate the transfer of water by bulk due to high addition. Water molecules are attached to the cellulose molecules in the xylem wall. Cohesion of water molecules is usually high due to cohesion of water molecule is unusually high. It doesn't say high any cohesion of Cohesion of water molecule is unusually high due to hydrogen bonds among water molecules. Therefore, continuous water column is formed within the xylem vessels and the tracheids. Addition and cohesion facilitate transport of water by bulk flow. Due to high addition, water molecules are attached to cellulose molecules in the xylem wall. So, weak bonds formed between the water molecules and the xylem vessel walls help to maintain water column within the xylem. I don't see any error in it, right? I don't see any error in it, right? It's perfect. So why do you choose fifth one for the during phloem unloading, sugar movement towards the sink as well as water movement towards the xylem from the phloem through osmosis. During phloem unloading, sugar moved towards the sink as well as water moved towards the xylem from phloem occurs through osmosis. Water movement occur through osmosis, right? Water movement occur through osmosis. Doesn't this give a meaning, people, that the sugar also moved by osmosis? I felt like that. That's why I took fifth one as the answer. During phloem unloading, sugar moved towards the sink as well as the water movement towards the xylem. Shall we check that statement? What the resource book says about it? It says, as a result of concentration, sugar molecules diffuse from the phloem into the sink and water follows by. So that diffusion word is missing. Diffusion word is missing, right? Sugar by towards the sink as well as, because of this missing word diffusion. Right? We don't have any other issues with the other statements, right, people? Do you have any issues with the other statements? I don't have. Okay, so I'm going to settle with that. Right? They have to mention, this gives like a meaning that the both events osmosis participate towards sink by diffusion. Sugar move by diffusion as well as water move by osmosis is correct. Right? That is my disagreement there. Question number 15. Question number 15. The incorrect statement is xylem vessel are absent in softwood. In dicot plants, cortex of the root is wider than the cortex of the stem. They are asking incorrect, ne? Dicot plant, cortex of the root is wider. Dicot plant, cortex of the root is wider. Cortex of the root is wider than the cortex of the stem. That is correct, ne, bhai? In the dicot plant, there is root does not contain people a pith. Pith absent mean the cortex is going to be thicker. Right? 15th, they are asking incorrect statement. Right? Incorrect statement. Xylem vessels are absent in softwood. Check this one. What is softwood? The gymnosperm wood. So this statement is correct. Dicot plant cortex is wider. Correct. Vernonia stem. Monocotymbia, Vernonia stem section stained with aniline sulfate can be used to examine secondary vascular tissues. I think it's correct because people, this is Vernonia. Monocotymbia, මොකටද ගත්තේ? Lignified secondary grown or primary grown one? I think primary grown we took people Tradescantia. Secondary grown we have taken people මොකද්ද ගත්තේ? Secondary grown. Monocotymbia ගත්තා. Helianthus also we took, ne? Helianthus, right? So therefore, I think that statement is okay. Fourth one, we will with time, the cocambium breaks and new co-cambium form from outside of it. No, inside, neither. Outside can a word not fit. Right? So outside, inside. Many parenchyma cells are totipotent. That is okay. Ne? So I'm going to take fourth one as the answer. Then people, question number 16, the correct statement is, question number 16, correct statement is, the Neplephis gametophyte rhizoids develop into, in Neplephis gametophyte rhizoids develop on the ventral side of the antheridium. Neplephis gametophyte rhizoid develop on the ventral side and the antheria develops on the dorsal. Wrong. Both the gametophyte rhizoid and the antheridia, archegonia, all are on the ventral side. Wrong. All the development stages of the young sporophyte of both Pogonatum and Neplephis are nourished by the gametophyte. All the development stages of the young sporophyte of both Pogonatum and Neplephis are nourished by the gametophyte. Not wrong neither. I feel it's okay. When the genetic content of the ovum is duplicated, when the genetic content of the ovum is duplicated during parthenogenesis, always fertile seeds are developed without fertilization. When the genetic content of the ovum is duplicated. What is the word they use, people? Right? When the genetic content of the ovum is duplicated, when the genetic content of the ovum is duplicated, when the genetic content of the ovum is duplicated during the parthenogenesis, always fertile seeds are developed. Always fertile seeds are developed. I think we have to check that word always. Develop without fertilization. Right? What it says, people? Always, sometimes, most of the time. What is it? And if flowering plants has not been pollinated, only the petals will fall away. If the flower has not been pollinated, only the petals will fall away. Wrong, neither. Some of the petals are going to fall, people, off uh, even after fertilization, because they are not permanent parts. So they are falling away anyway. Wrong. And then the forest canopy absorb most of the far red anyway. Absorb far red red light. No. Only the red light is going to pass through. So what is your answer for this? What is your answer, people? Do you agree with the second statement? Right? When the genetic content of the ovum is duplicate during the pathogenesis, always. Shall we check that part, people? Always. Always. Sometimes, most of the time. What is given? Life cycle parthenogenesis. Let me read it for you. In parthenogenesis, the genetic content of the duplicate to become diploid, enabling the genetic content of the egg is duplicated to become diploid, enabling the seed development without fertilization by a sperm. When the genetic content of the ovum is duplicated during parthenogenesis. Now, parthenogenesis can occur in three ways. Parthenogenesis can occur in three ways. Egg is resulted by mitosis and hence diploid. Haploid, the ovum fuses with the polar nuclei. Right? And then genetic content of the egg duplicate. Right? When genetic content of the ovum is duplicate during parthenogenesis, always a fertile seed are developed without fertilization. I think this always word is introducing an issue here. We can't agree with that because it's not mentioned like that. But anyway, pathogenesis can occur in three ways, but it doesn't fit. So I think I can settle with the second one as the answer. If you have any arguments, we'll see that. Question number 17 in plants. Question number 17 in plants. Wilting of leaves. I don't have any arguments here. So we'll take people second one as the answer. You check your notes. Question number 18, people, the correct statement regarding genetic content of the tissue of the body is. Correct statement regarding the, general connective tissues of the body is. General connective tissues of the body. What is your answer, people? Contain three major types of cells, connective tissues. Contain three major types of cells. We know WBC is present, right? And fibroblasts are there. Fat cells are present. So we can think of three main cell types, but I think it's not mentioned. Three major types of cells are there. I think the white blood cells, fat cells, fibroblast. Um, what are the cell types given? Let me read that out. Connective tissues. They say, people, um, different types of cells can be found in the matrix, such as fibroblast, macrophage, and mast cells. In addition, fats and leukocytes are found. So they are giving about 5 cell types, right? About 5 cell types they have mentioned. So I think we can disagree here. Densely packed without collagen fibers. Densely packed without collagen fibers. Wrong, neither. Fibrous tissues are densely packed, wrong. Contain fat cells with large fat droplets. That is correct. Fat cells are there. Found within human vertebral column. No. Vertebral column, no. We know, people, vertebral column inside some of the marrow tissues are there. Um, vertebral column. Then, people, unique only to mammals. Which one? Connective tissues are unique to mammals, wrong, neither. So how about taking 3, right? Densely packed with collagen, no. Found within human vertebral column. Vertebral column. Connective tissues are there, even bones we take as connective tissues, cartilages, connective tissue. But they say found within human vertebral column. So, oh, no, neither. So I'm going with the third one. I think you agree with me, right? I think you agree with me. Question number 19. Question 19, people, the correct statement regarding the correct statement regarding regulation of digestion in man is. Correct statement regarding regulation of digestion in man is. Digestion by nervous regulation starts within the stomach. Nervous regulation. Isn't it mouth, people? Salivation and all? Wrong, neither. Secretion of gastrin is stimulated by the secretion of gastric juice in the stomach. Gastrin is stimulated by the gastric juice? No. It's actually the mechanical stimulation. When the food touches the inner lining, it starts secreting it. And then when the chyme is rich in fat, it inhibits the gastric juice secretion. Chyme is rich in fat. It inhibits the gastric juice secretion. I think I can agree with that. Peristalsis in the stomach is inhibited. Peristalsis in the stomach is inhibited only by cholecystokinin. Wrong. Why? Secretin too. And then cholecystokinin and secretin act on the pancreas and stimulate the release of digestive enzyme. Cholecystokinin and secretin act on the pancreas and stimulate the release of no digestive. You know, people, secretin is releasing bicarbonate. Secretin releases bicarbonate, so we can't agree with that. So I have the answer now. I have the answer. Let me take third one as the answer. Question number 20. Question number 20. Which of the following statement regarding human respiratory system is false? Human respiratory system is false. Surfactant reduces the surface tension of alveoli. Surfactant reduces the surface tension of alveoli. Surfactant reduces the surface tension of the alveoli. Correct. Larynx and the trachea are entirely cartilaginous. Larynx and trachea are entirely cartilaginous. Wrong, Carbon dioxide partial pressure and oxygen partial pressure of blood leaving the alveoli. And those of the alveoli AI is equal. Correct. The partial, we discussed that recently, last week. The partial pressure of carbon dioxide and partial pressure of oxygen and partial pressure of carbon dioxide in the body tissues are lower. The partial pressure of oxygen lower, carbon dioxide higher respectively. Correct. Nicotine in cigarette smoke temporarily increases the blood pressure. Correct. But we didn't discuss this part yet. We'll finish it this week. Right? So I'm having the answer now. Second one is my answer. I think you can agree with me. Hari. Then we have to move to the structured part. Lamai, shall we move to the structured part? What's the time now? Silly. The time. 9:30. 9:30. All right. So let's quickly go through this. Could you please tell me the answer for the sixth question? Sixth question. Oops. Sixth question. We have taken it fourth one as the answer. Resource book says the saliva, releasing of saliva to the oral cavity occur when food enters the oral cavity due to nervous reflex. So what? So what? So what? So can I get the answer for the 15th and the 16th? 15th and 15th 4, 16th second. 15th 4, 16th second. All right. Okay, now moving people to the structured questions. Right, moving to the structured questions. So let's complete that. Let's do it quickly. This one apparatus, the following apparatus is constructed by a student to determine the rate of the transpiration from the leaves and shoots of a plant. What is the name of this one? What is the name of this apparatus? What is the name of this apparatus? Ganong spotometer. Oops. Ganong spotometer. Name the parts as A, B, C, D. What is that? People twitch of coleus plant. Twitch. Twitch of coleus plant. Coleus plant. You can just write twitch, but better twitch of coleus plant. What is B? Rubber stopper. Rubber stopper. What is E? E is beaker of colored water. B, C. What is C? Tap. D. D is graduated capillary tube. Graduated capillary tube. Graduated capillary tube. E. Beaker of colored water. Beaker of colored water. Colored water. Beaker of colored water. F. Stand. F is a stand. Next one. Next question, people. How is the air bubble X is introduced into the apparatus? Now you write there, lift the free end of the capillary tube. What are you going to write? Lift the free end of the capillary tube. Right? Lift the free end of capillary tube. Lift the free end of the capillary tube. Allow air bubble to enter. Allow air bubble to enter. Allow air bubble to enter. And immerse the free end back in beaker. Immerse the free end back in beaker. All right. Okay. Then we are moving to next one, people. What reading should be, should the student take to obtain the rate of transpiration? How to obtain the rate of transpiration? Time taken for the air bubble to travel a particular distance. Time taken for air bubble. Time taken for air bubble to travel particular distance. Particular distance. State the two major types of circulatory systems in animal kingdom. Two major types of circulatory systems in animal kingdom. Open circulation. Second one is closed circulation. Write down two risk factors for hypertension. Right? First one is obesity. Next one, people, diabetes. Mellitus. Family history. Smoking. Smoking. And then you can take high intake of salt. High intake of salt. And you can write a few points like that. What is lymph, people? Lost fluid from blood capillaries. Lost fluid from blood capillaries when they inside the lymphatic system. When they inside lymphatic system. When they inside lymphatic system. Right? Okay, next one, people, they want us to write a draw flow diagram to show how water insoluble products are absorbed and then transported into the circulatory system during fat digestion. Right? Draw a flow diagram to show how water insoluble products are absorbed and then transported into the circulatory system during fat digestion. What are you going to write, people? You'll see that fatty acid plus monoglyceride. Fatty acid monoglyceride go into epithelial cells, ne? Epithelial cells. And then what happened now? Triglycerides reform. triglycerides are going to reform. And then chylomicrons are going to form. Chylomicrons are going to form. And these chylomicrons are now entering into lacteal. Chylomicrons go into lacteal. From the lacteal, it goes into lymphatic vessel. Lymphatic vessels. And then it goes into blood. It goes into blood, right? That's the path. That is the path. All right, and then people move to the last task. Explain the significant features of seedless vascular plants. Significant features of seedless vascular plants. Right, seedless vascular plants. Significant features of seedless vascular plants. Now, you know, people, seedless vascular plants, there are some features. What are those features present in seedless vascular plant? Can you remember, people? Seedless vascular plants. Helicobacter pylori, why? Was there a question regarding that? Seedless vascular plants will, we said that there were several features. What are the features, people? Seedless vascular plants. Come on, tell me. We said, people, seedless vascular plants. They had roots, leaves, vascular tissues. They were developed. So you have to write that part, right? Seedless vascular plants during the evolution, they had evolved stem. So you missed 3 Lua. Okay. Did I? Okay. Okay. Then you know the answer. 3 third part. Where is it? Third one. Name the bacterium. Okay. All right. So my fault. So seedless vascular plants, we said, people, in evolution, they evolved these characters. So you write that, people, develop the xylem and the plant. Significant features are seedless. Transportation through the xylem and the plant. So you give a small description on that. Then evolution of roots, evolution of leaves. So those 3 features you must write. And then describe the mechanism which guttation occur. And that's a good question. Guttation, how the guttation occur? You know, people, transpiration fail in the night under high humid condition. As a result, the water lost by transpiration is less, so that the volume of water which enter the leaf is greater than the volume of water lost by the transpiration. Because of that, the positive pressure build up, and then this is now resulting, people, the exudation of this water and minerals through the tiny pores present in the leaf margin. What do you call the hydrothodes, right? So that part. So root pressure and guttation. So I think you have to describe that. I'll fix a marking scheme on that. I didn't have enough time to do that. I'll fix a marking scheme and send that. So anyway, write that as a question, right? And that's all. So could you increase the number of questions regarding digestive system? And next one, the provincial paper, the guttation was asked to write short note. Good, right? Yeah, now we are starting a paper class from January onwards, right? We start the paper class, so we'll do a full paper every week, Wednesday. You can join online, right? So we'll do more questions, don't worry, right? So you vote for justice, no? Justice. You know, right? You know, right? Justice, that's all. Okay then, see y'all. Right, see y'all. Right, jai ve bye.